Welcome to the CP, and we are discussing the AIPMT main solution. And next question is question number 50. Tobacco plant resistant to a nematode have been developed by introduction of DNA that produce in the host cell. In this, we use the tobacco plant resistant by the nematodes, and in this, roots are infected with these nematodes, like Melacogna incognita, like this nematode affect the root. So we use transgenic bacteria, and in transgenic bacteria, a DNA is present, and that DNA, when the nematode feed that transgenic bacteria, I am explaining this with this diagram. When this bacteria, transgenic bacteria, we use this transgenic bacteria, in this double strand DNA is present. That DNA form is both the strand of this DNA, sense and antisense, both this form RNA, both these strand form RNA complementary. And when this nematode feed on this bacteria, transgenic bacteria, which contain a specific gene, when this enter into the intestine of this, and this enter into its gonads, and in this gonads, this genes suppress its mRNA and its cells. So this kill this gonadal cells, and that destroy the nematodes present in the roots of tobacco plant. And by the suppression of this gonadal cell, this kill the nematodes, and by the killing of this nematode, this destroy this nematode, and so we produce the tobacco plant resistant to the nematodes by this transgenic bacteria. In this transgenic bacteria, this type of DNA is present. The DNA form, both the strand of this DNA form sense and antisense strand, both the strand form the RNA, double strand RNA. And in these cells of this gonad, we will see this phenomena like this. This double strand DNA enter into it, and by the dicer protein, this cut down into pieces, and after the protein is attached with it, and this form the RISC complex, RNA induced silencing complex, and this turn into single stranded complex, and single stranded complex, this contain overhang sequence of RNA. This is double strand RNA. This double strand RNA is formed by the gene that introduced in its DNA, and that gene which form this RNA, and now this double strand RNA form this complex single strand overhang sequences, and this destroy the mRNA produced by this DNA, like this DNA produce this mRNA, and this sequence is now attached with this sequences by the overhang sequences. And this sequences, this protein exonuclease enzyme, and cut down this into several pieces. So this mRNA is destroyed, lysis, and this is called gene silencing. And so this gene which enter into this gonadal cell produce both type of double strand RNA. By the sense or antisense strand, both the strand form this double strand RNA, and that destroy this mRNA of the cell. And by this expression of this cell is stopped, and this kill the gonadal cell. So the right answer is fourth. This gene form both type of sense and antisense strand. Thank you.